And I will then proceed to our final speaker of the day. I want to introduce Dr. Dae Kwan Ko. Dae is an assistant professor at Michigan State University, where he's investigating how genes are dynamically regulated in plants in response to climate change using multi-omics network approaches. And today he's going to speak to us about decoding abiotic stress resilience in sorghum, a transcriptomic framework for climate ready crops. So without further ado, thank you for we can see
Okay,
your
that's
slides.
okay. You can see it.
Yes, we can see the slides, we can hear you.
Sounds great. So thank you for the kind introduction. I also like to thank for the opportunity to present the first WALII Symposium, and I've learned a lot about different stuff like desiccation, many others. And now I'm very excited to share my research, which is funded by the Great Lakes Bioenergy Research Centers. My research is not related to desiccation, but the approach that I'm taking is highly applicable to, I think, many of your research. So let's get started. So let me begin with one of the principal concepts, or fundamental principle in biology. So in DNA, even though DNA contains enormous of information in any organisms, only specific subset of the gene is required for specific function and time and tissue, environmental condition. They are transcribed into mRNA. So the expression is highly dynamic, not static, highly dynamic. And most importantly, in virtually any organism, gene expression changes serve as a first responder to virtually all biological process. So my research is more like involved in what gene regulations, how gene regulation is working in this biological process. So mechanistic level, gene regulation highly depends on interactions between DNA and the protein called cis-regulatory element or transacting element. So cis-regulatory element or cis element, a short DNA sequence in gene promoters. And transacting elements, mostly transcription factors, the protein encoded somewhere else in the genome, then bind this cis-regulatory elements. For example, here, the gene A has specific DNA sequence which matches binding site of this TF1. So TF1 can bind the sequence and gene A can be expressed when required. On the other hand, gene B has different sequence. The TF1 cannot bind this gene B. The gene B cannot be turned on. So this simple example highlights the key concepts. The DNA sequence embedded within promoters largely determine which TFs can bind, ultimately when or where genes are turned on and off. But interestingly, DNA and protein, the transfector, they don't work in isolation. They work in kind of like our social network in our lives. The transfector TFs form extensive network with the gene they regulate. So so if we understand the gene regulatory gene networks, this if we understand gene function in the network level, not a single gene or protein, we can better understand because this network is enabled the complex organisms like a plant to coordinate biological responses more efficiently and more integrated way. So this concept has kind of similar like an analogy from this book. The concept described this book, The Wisdom of Crowds. So what's saying is that when information is aggregated across many individuals, the collective insight often surpasses the contribution of any single members. So network biology leverage the collective behavior of many regulatory interactions, not a single protein or gene, to reveal the principle that will be invisible when examining gene or TFs one at a time. So in my research, we had questioned then how can we harness this gene regulatory dynamics and network biology to enhance crop resilience. The urgency of this question becomes clear when we consider climate stress as a growing threat to crop productivity. This graph showing the temperature fluctuation y-axis and sorghum yield on x-axis. By the way, sorghum is a major crop in the primary biofuel feedstock in the United States. So it's very important economic value as well. So you can see So even though sorghum yield has been steadily, gradually increased, there's some recent temperature change makes really dramatic negative impact. For example, here, the past 2 decades, rising temperature over the past 2 decades strongly correlate with the reduced sorghum productivity. So this example highly underscores the urgent need to elucidate molecular mechanism underlying crop resilience to environmental stresses. So in this project, to understand how plant reprogram gene expression under different abiotic stresses, so we generate a comprehensive RNA sequencing data set. So we profile shoot to root separately and 4 different time points or key time points based on the literatures and drought, heat, salinity stresses. In this design, we are able to identify how these three factors, tissue identity, time after stress application, stress type, get together, shaping transcriptomic changes in sorghum. So this principal component analysis shows a striking, very clear pattern, which is tissue identity dominate the transcriptomic variation. Like you can see here, the root sample and the shoot sample, they separate each other, right? And that's indicating fundamental different roles in stress perception signaling between root and shoot. Even each tissue's root or shoot, the sample that cluster by time point, then stress type. So as you imagine, the drought, the heat, salinity, they all connect to each other molecular level. Gene that responsible to stress, drought, they may be responsible to salinity stress for different level maybe. So this kind of expected. So what we found out, the tissue identity is dominant factor than time and stress type. So that indicates the plants activate the shared regulatory programs across different abiotic stresses. In the meantime, the stress tissue-specific regulatory architecture plays much larger role in shaping the overall transcriptome. So even though the stress type contribute to less variation than tissue type or the time point, this stress-specific expression pattern also have some biologically meaningful data. For example, here, this gene encodes oxygenase superfamily protein. This gene specifically induces salinity stress late phase in the shoot. But in root, this gene encodes matchbox transfactors associated with root cell differentiation based on an annotation of different patterns. It's highly robustly induced heat stresses and compared to the drought and salinity stresses. So we have all this information. We perform now co-expression network analysis to identify stress responsive gene set tissue. This approach is basically just group the gene based on the expression pattern across dozens of these tissue sample types. So we got a lot of different modules, imagine, but we have a very interesting kind of module here is shoot and root separately. And this module have similar number of genes, similar number of sets, number of genes, and they have no expression change much in other stress, but they have strongly downregulate the middle phase of heat stresses. And interestingly, those genes are not overlap much. Only few genes that overlap between these two different modules, two different tissues, even though they have similar expression patterns. And then in line with that, they have different biological pathway involved in the energy metabolic pathway in each this shoot module, oxidative stress in the module found in the root. They're indicating the tissue-specific nature of heat-induced transcriptional reprogramming. So next question to identify kind of architecture of this regulatory gene network in this module, we assess this kind of network connectivities through metrics like centrality, which indicate how close a node is to this gene, how close the gene is to all other genes, and the degree, which is number of co-expression interactions. First, what I found is a lot of hormone-responsive genes that highly ranked in the network. They're indicating this hormone-responsive signalings that tightly link these heat-specific middle phase suppression during the stresses. Then based on these numbers, and I was able to pinpoint the top ranked gene. And the beauty of the co-expression network analysis is that not just giving you kind of group of genes that have co-expression pattern, but also give you the metrics. So you can quantify the trait or network of trait. So based on that, I was able to pinpoint the most important gene I just called regulatory hub genes. The next question is then which transcription factor could regulate these regulatory hub genes? And the idea is if we identify this transcription factor, then we can manipulate this most important gene for this stress resilience. So like to identify that, we perform the gene regulatory network modeling. So the long story short, with the gene regulatory network modeling, which is based on the expression dynamics, we have multiple different samples in each module. I was able to pinpoint top three ranked TFs. And for the gene, we pinpoint the regulatory hub genes. And, and to test and identify the validate these top ranked transfectors for potential candidate for regulating these regulatory hub genes. So we leverage evolutionary conservation of seasonal regulatory elements in maize. The idea is if the DNA sequence is regulatory elements is important, must be conserved from two different species. So maize, sorghum is highly closely related. So the comparative analysis is kind of a powerful mean to identify whether this sequence is highly important for the evolution perspective. So what I did was found homologs of ESP11, major homologs. Then I have four homologs. Three is highly matched, 95, more than 95%, something like that. The other one is 80%. Then they perform the scanning of these promoters, which means I just blast with this sequence against the sorghum promoters. What I found is specific the region of few hundred base pair region highly conserved on these highly closely related major homolog genes, but not the one is distantly related. Importantly, this region contains B binding motifs, which support our prediction that B binding among the top three B transfer factor could regulate this target genes. For the STH25, the other regulatory hub gene, the other root module, we found specific two regions, promoter regions, highly conserved. One is closely related to major homologs, but not the one distantly related. This region importantly have B3 WALII transfectors, which is exactly matched our prediction that WALII B3 could be the one that regulates this STH25, which is a regulatory hub gene, this module. So to wrap up, so today I'll show you Abiotic stress triggers the dynamic transfer reprogramming shaped by two major determinants, called tissue and time, the stress type. By analyzing all these three determinants within a single framework, I was able to identify tissue identity is a major one for regulating the stress-inducible gene expression in sorghum. So series of network modeling and cross-species promoter analysis that identified distinct shoot and root specific gene regulatory network, which are mainly organized by around key regulatory hub gene and their predicted upstream factors. The functional validation of these predicted regulators in the way will be essential to fully understand the regulatory dynamics and most importantly, translate this insight into strategy for engineering tissue-specific stress resilience. So with that, I'd like to thank Dr. Federica Brandigi, who is the PI for this project, also the past, the current research technician to provide the plan material. This work is supported by the DOE through GLBIC and JGI. And thank you for your attention. I'm happy to take any question.
Okay. So to start off the questions, Cataline asks, are these is your data published somewhere that where we can look at?
That's a great question. Yeah, it's on the way. It's under review, and we're thinking about putting bioarchive also. And yeah, so it'll be available very, very soon.
I had a question. So all of your work that you've presented is in sorghum, right?
Yeah.
So for I know that there's like I think actually I'm not 100% sure that there are more desiccation sensitive sorghum as well. Or just drought sensitive, actually, not desiccation. For those, have you seen some of these transcription factor also like in the same regions of the tissues? Do you expect to see those across species as well?
You know what? Through today's the symposium, I learned a lot about this desiccation, and I realized a lot of molecular factors involved in desiccation, a lot of transcript factors or a lot of metabolic gene. I'm definitely interested in looking at those genes in my data set, many others also. Yeah, there should be the connection between sorghum and desiccation species gene. Yeah.
Gotcha. Yeah. Um, yeah, and I think if anyone else has any questions, please feel free to put it in the Q&A or in the chat for um, Dae Kwan to get back to you later as well.